Hello and welcome to another episode of Carbohydrate Discussion Series. Today, Abhishek will present assessing O-Naphthylmethyl and O-Anthracenemethyl Glycosides as metabolic inhibitors of bacterial glycan biosynthesis. Let's begin the session.
Assessing O-Naphthylmethyl and O-Anthracenemethyl glycosides as metabolic inhibitors of bacterial glycan biosynthesis. The abstract of this paper, bacterial glycans play a crucial role in survival and pathogenesis, making them attractive antibiotic targets. Unlike mammalian glycans, bacterial bacterial glycans incorporate sugars such as bacillocamines and acetyl-fucosamines and 2,4-diacetoamido 2,4,6-trideoxygalactose. To probe the role of bacterial glycans, we we previously developed O-benzyl glycoside that metabolically inhibit Helicobacter pylori glycal biosynthesis and impair bacterial fitness. Here we prove the efficacy of O-naphthylmethyl and O-anthracenemethyl glycosides, which bear larger aglycons relative to previously reported bacterial metabolic inhibitors. In this paper, O-naphthylmethyl D-N-acetyl-fucosamine inhibited H. pylori glycan biosynthesis, reduced biofilm formation, and impeded H. pylori growth at lower concentration than its O-benzyl analog, while leaving glycosylation of the commensal bacteroides fragile intact. This Helicobacter pylori, H. pylori, is a type of bacteria that infect the stomach and is a common cause of peptic ulcer and gastritis. These are the previous reported molecule, that is deoxyamino monosaccharides found in bacterial glycans. That's this BAC1, that DG2 and FUCNAC3. These are the reported metabolic inhibitors, that is 4 and 5, and these are the these represent substrate decoy mechanism of inhibition. Coming to this work, in this paper, assessing O-naphthylmethyl and O-anthracenemethyl glycoside as a putative bacterial metabolic glycan inhibitor. As we can see in the left-hand side, these 6, 7, and 8, these are the naphthyl derivative molecules, and in the right-hand side, this 9, 10, and 11, these are the anthracene derivative molecules. These are the novel O-naphthylmethyl and O-anthracenemethyl glycoside inhibitor introduced in this study as a putative metabolic inhibitor designed to disrupt bacterial glycan biosynthesis. Coming to the synthesis of these molecules, how this O-naphthylmethyl and O-anthracemethyl glycoside derived. Starting from this thioglycoside that is reported by William et al. In this paper, after 6 steps, up to 40% yield, this C2 and C4 diazido compound formed. Then by taking these molecules in the presence of NBS, THF is to S2O for 15 for the reaction time is at 15 minutes, converted into hemiacetal, and then by the reaction with trichloroacetamide converted into TCA donor, then taking as an acceptor naphthyl acceptor, did the glycosylation reaction to form this glycoside product, that is with the alpha is to beta 2:1 ratio. And further, this azido is going to reduce to NaCC in the presence of zinc acetic acid. Then after reaction with acetic anhydride, the acetylation is going to take place to form the required compound, this 6 molecule C2, C3, and acetyl. Then taking from that same starting molecules in the 3 step, what he did is that only change the epimerization of C4OH to see this azido compound, and then further same scheme is followed, is converted into hemiester, then further converted TCA donor, then taking as a naphthyl molecule as an acceptor to form these 17 molecules. Then further he reduced in the presence of zinc acetic acid and this and did the reaction with acetic anhydride to form these 7 molecules. And in the last, what he did is that only convert C2 into this azido and follow the same reaction scheme. These are the synthesis of 9 anthracene analogues of this 9, 10, and 11 bacterial anthracene, dato anthracene, and FUCO anthracene molecules. He followed the same reaction scheme what he did in the case of naphthyl derivative molecules, starting from this 13 he converted into hemiester, then further converted into TCA donor and did the reaction with 9-anthracene methanol, taking as an acceptor to form this anthracene glycoside product in the ratio of alpha is to beta 1 is to 12. For reducing this entry into NSAC, he did several experiments by changing the reagent, as I'm going to discuss in the upcoming slide. Anthracene glycoside product. And these are the table for this table one for reducing entry to NHAC. He changed the several reagent starting from zinc acetic acid to the this PPS3, and he also did with the stannic chloride. In this row 8, he found 74% over two-step by using this PPS3 pyridine taking as a solvent THF is to H2O in 9 is to 1 at 60 degrees Celsius. Reaction time is 12 hours, and then the reaction with acetic anhydride to form corresponding this 9 molecule in alpha is to beta 1 is to 14 ratio. And how this molecule is starting from 12 to 13, 12 to 14, 12 to 15 can be synthesized. I'm going to discuss in this slide, taking as a starting material as in this thioglycoside product. Reaction with triflic anhydride in the presence of pyridine, then using this TBA-N3 as a nucleophile to form this 12 molecule, this 12 compound. Then further reaction with triflic anhydride pyridine, same in the presence of the DCM as a solvent, and taking sodium azide as a nucleophile to form this C2 and C3 diazido compound. Then this thio is converted into hemiester, and this thio is activated by NIS and did the glycosylation in the taking as an benzyl alcohol to form the 14 molecule to form this 14 compound, and further it is going to be reduced by zinc and protected by acetic anhydride. These are the synthesis of this DAT DG Dynamic OBN. Starting from this 11, again he did the experiment. He did reaction with triflic anhydride, then reaction with sodium azide to form this epimerization of C4 into C4OH into C4 azide. Then the glycosylation as it activates the sulfur is activated by the NIS and it glycosylation with the taking as a benzyl alcohol as an acceptor to form 16, then further reduced and protected by acetic anhydride. these are the same. He formed these three molecules following the same reaction scheme. And these are the synthesis of 3F bacteriodynamic. This these fluorine derivatives molecules, as he follow the same reaction scheme as I discussed earlier. In this paper, he did this experiment that is called Western blot analysis. As we can see in this slide that these are the, in left hand side, these are the O-Naphthylmethyl glycoside that disrupt H. pylori glycans, and this right hand side, this O-Anthracenemethyl glycoside, and that has been, sorry.
Continue. I couldn't see the Western blot. Maybe that's some technical problem. Continue.
Okay. In left hand side, these are the O-Naphthylmethyl glycoside that disrupt H. pylori glycans, and this right hand side, this O-Anthracenemethyl glycoside that has a minimal effect. As we can compare this to plot, there is no any changes seems to appear. In the case of and these are the molecules 6, 7, 8, and these are the anthracene molecules 9, 10, 11. He did experiment with these molecules. And what he did that by taking these naphthyl derivatives and anthracene derivatives molecules, he found that in the case of naphthyl derivatives that has a that disrupt the H. pylori glycans. So what he did is that by taking this FUC NAC O-Naphthyl, by lowering the concentration 0.25 to 0.01 to 0.25, he did again experiment. Yeah, and he mentioned that this FUC NAC O-Naphtylate was further evaluated at lower concentration, 0.01 millimolar to 0.05 millimolar, and 0.1 millimolar, and at 0.25 millimolar, to assess its inhibitory effect on glycan biosynthesis. This AC4 served as a positive control, and a treatment with the azide-free sugar AC4 glucanase AZ treatment with no inhibitor additions, and this glucanac AC served as a negative control. And this right hand side, these are the changes in the surface glycan profile. This is a flow cytometry analysis demonstrated a change in the fluorescence intensity of Corne upon treatment with 1 millimolar bacterionaphthyl 6, this 1 millimolar datonaphthyl 7, and this 1.1 millimolar this FUC NAC O-Naphthyl 8, compared to the untreated sample, consistent with the perturbed cell sulfur glycan profile. And Corne pre-treatment with 400 millimolar mannose carboblock prior to probing untreated H. pylori led to decreased binding. As we can see in this graph that these are the this gray color is untreated, and by comparing with this, this bacterionaphthyl, datonaphthyl, and this FUC NAC O-Naphthyl have a more fluorescence as compared to this untreated molecule. And this conclusion of this paper, the O-Naphthylmethyl glycoside derivatives, this FUC NAC O-Naphtylate, exhibit a potent inhibition of a glycol biosynthesis in H. pylori, demonstrating observable effect at the concentration lower than those of the previous tested inhibitors. FUC NAC O-Naphtylate induce multiple bacterial fitness defects as evidenced by a significant reduction in biofilm formation and impaired growth. Notably, this FUC NAC O-Naphthyl did not affect the glycan biosynthesis or fitness in the commensal bacterium Bacteroides fragilis, and suggesting its selectivity. By contrast, the analogous of O-Anthracenemethyl glycoside failed to exhibit a significant inhibition of H. pylori glycan biosynthesis. And this given that This aid effectively disrupt H. pylori protein glycosylation. It can serve as a chemical probe to investigate the functional role of glycoprotein in H. pylori. Thank you.
Hello.
Hello.
A nice presentation, Abhishek. You go to the title slide, sir.
Title slide.
Title slide, sir. So basically, these different substitute means sugars are there, na? So that sugars are usually there on the bacterial surface also, right? This glucose, bacillus, something are there, right? This is introduction. You are saying that unlike our mammalian bacteria have a different sugars like glucose and all.
Hmm.
So this kind of sugars already there on the bacterial surface, but whenever you attach naphthalene or anthracene, that time they act as a inhibitor.
Yes, yes.
So that is the whole concept of this one.
I think also this.
So that is the only means whatever they showed some Western blot, right? That is the only way they showed the inhibition of a bacteria. Like
Like
is there any other?
in this paper, several other experiment he also did, but I didn't understand too much, so that's why I take this only this Western blot analysis.
Okay, okay, only Western blot you took.
On the basis of this, how he select this 8 molecule that effectively uh, did this fusinec O-Naphthylmethyl molecule.
I don't know what they have exactly taken here means for analysis. So something they are seeing the difference compared to the
Other
whenever
molecule.
molecule. So based on I don't know what exactly
Like
that usually proteins antibodies are taken in Western blot. Maybe they might have taken bacteria also.
bacteria also means.
It's like usually cells also they'll do the Western blots. This bacteria also cells, right? So I don't know. I don't exactly know. But that can also be one of the reasons. Otherwise, they might be taking the glycoprotein, whatever that expresses on the surface of the bacteria. That glycoprotein is then they spray it. So, if in case if possible, that if paper is open, that can you open that paper? That is,
That paper.
let's see. Uh, if it is open,
It's visible.
go below.
Like here he did this experiment also, this analysis, sorry.
Which analysis?
Like
I
this
don't
one.
see it. Okay. Oh, okay, okay. Now I can see. It is very much
I
blur.
think this is bacterial inhibition experiment, right? They have done here.
Here, right.
It is very much blur here. Maybe the network problem mine is. Okay, that's the analysis. That is. So, primary growth. Okay, 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 okay. Like we do MTT and all. Somehow. How? Uh, this is Western blot. Mhm. Okay, okay. You could have shown that one also, like the bacterial inhibition studies are there, na? means
Yes,
since
yes.
since you are telling that you couldn't understand much of it, that's fine only. Okay, thank you.
Okay, thank you.
You go to your glycosylation slides you have shown earlier. Like if you will see in all the places they are using different solvent. Mostly they have used acetonitrile for glycosylation.
Yes,
In
yes.
some of the places DCM also. Like here they are using dichloromethyl. The previous I think. So that they have changed because of the solubility or something else. Like here they have used acetonitrile. And still they are not getting, I think. So here they are doing for selectivity or solubility also has some role.
Like
Like
like if you see in the case of naphthyl derivative, he go
the,
with DCM as a solvent, and here he find this alpha into beta, uh, like more alpha product. Like if you see that 17 and 18, this
yeah.
alpha, but in this case, here like both are forming, but beta is more.
So did they
And
mention they are doing this to get the selective product, stereo selective, or because of solubility they are changing the solvent? They might have mentioned some.
They have mentioned, but I didn't saw. Maybe because of like solubility. What
Because
I
you were
think that.
also facing similar problems, so
Uh,
maybe
that's why
solubility had some role.
solubility is one of the reason I think.
That could be one thing you can check.
Good presentation, Abhishek.
Yes, thank you.
Why they are changing the thioglycoside to first TCA and then doing glycosylation?
Here,
Hmm.
like he in this paper he's starting from this molecule. Like this, these are the starting molecules.
No, but
I
we
don't.
can just activate
Uh,
directly.
like if there is a hemiaster, then we can go directly for TC and did the glycosylation.
No,
But
in this also we can directly activate this thio, then we can.
maybe maybe that with the like I also did this experiment now. So in the case of thio, it's not working properly. That's why I also go with the TCA donor. Maybe he also faces that problem. Like he converted into hemiester and make the TCA, and with the TCA donor, that glycosylation is working um, smoothly. That's why in the all, if you see in the all molecule, they convert in, they make the donor TCA and then go with glycosylation.
Or any selectivity issue.
Huh?
Selectivity, alpha beta.
Selectivity issue.
Uh,
What I think if you shall see this benzyl group, they are doing with thio only. They are using
With high
ester.
only.
But the moment they are moving to anthracene or naphthyl, maybe the promoter system is not orthogonal with the acceptor. Maybe it is getting reacted with the acceptor or something, some side product may be forming. That's why they are converting into the TCA donors. So in that case, it won't react, it won't do anything to the acceptor. Probably
oh.
that will be the case. Because Abhishek also has similar problem. So in that case, NIS was doing some reaction with the anthracene or naphthyl group. Because if you'll see benzyl, in that case, they have coupled benzyl group directly in thiodonor.
Directly, in NIS, by taking it as
They are
NIS and
changing,
taking.
so they are changing this donor and constantly they are changing this solvent system also. Different cases.
Yes, yes.
Okay, Abhishek.
Okay, thank you.
If you enjoyed this session, don't forget to like, share, and subscribe. Thank you.